Hi, I'm Dan Sands, CEO of True Concepts Medical. Our product is a 4-in-1 blood collection device designed to prevent false positives, false negatives, and process contamination to isolate pure uncontaminated blood to accelerate testing and diagnosis of sepsis. Unfortunately, every 3 seconds somebody dies of sepsis. There are over 40 million patients in the US that present to the ED with an unknown source of fever, which most physicians will determine and draw blood cultures to see if they're septic. So the gold standard is to draw blood cultures. So in that process, nearly 72 million people annually present to the ED and have their blood drawn for cultures. Unfortunately, what happens is during that process, contamination causes high false positive rate. So the contamination from the venipuncture, the skin flora, any subsurface bacteria, it's been proven to cause a high false positive rate. And that problem of if it comes back false positive, patients are admitted to the hospital, given unnecessary antibiotics, and which leads to unnecessary costs in our healthcare system to the tune of $8.3 billion annually. So the solution is technology. It's been proven that if you divert the initial contamination, called the initial diversion technique, has been proven to significantly reduce false positives. And now the CDC is recommending this initial diversion specimen technique as a best practice in healthcare settings. So this typically is used in the emergency department or ICU. The solution, Devia, is the first internationally patented 4-in-1 blood collection device, and it prevents false positives, false negatives, and also process contamination by isolating pure uncontaminated blood so that we accelerate that testing and diagnosis of sepsis. So the solution, how it works, it was invented by a 27-year veteran nurse practicing today as an emergency pediatric nurse. So the solution, you've seen multiple errors at the bedside. This solution diverts that initial blood draw through a vacutainer, which most people are familiar with. It's housed in this device, so the process would go venipuncture. This button is pressed. It will draw up the first 3 milliliter of blood, which is a contaminated blood, and then we draw up 20 milliliters of clean, uncontaminated blood. This contaminated blood will be sent off for procalcitonin, which is indicative of sepsis, while they're performing the other culture tests. So one of the blood culture bottles. So then the other problem is false negatives. So false negatives are caused by a high rate of underfilling of the culture bottles, which can lead to a false negative. False negatives are significantly bad because you're going to be sent home and you actually are septic, and this disease progresses rapidly and it could cause serious implications. So this device is graduated so you can see 10 mLs, 8 to 10 mLs is recommended for each blood culture bottle. So each is dispensed 10 ml and then 10 ml. So the clinician sees exactly 8 to 10 mLs going into that culture bottle to prevent false negatives as well. So our uniqueness in this space, there are two companies in the market, and it's early, but this is looks to be a technique that is moving towards a standard of care, and this technology is very innovative. It's streamlined and can be completed in the current workflow that clinicians and nurses are used to doing. So that the design of this allows for other biomarkers to be analyzed concurrently instead of wasting the blood. Other solutions on the market today waste that initial diverted contaminated blood. We repurpose it. So if you think of the 72 million blood draws annually, that's a lot of wasted blood, especially in pediatric situations. You want to draw the minimal amount of blood. So that's, again, a unique factor of our design. So first time visiting and attending the LSI event after multiple years of seeing the growth of the event. It's great to be here, and it's been excellent networking and speaking with multiple investors about our technology.